Let us pass on to the next part of classical genetics. What Mendel did the experiment, dihybrid cross. What is dihybrid cross? So it is a genetic cross which involves two individuals. The two individuals are differing in two characters. Or we can say, for example, it is nothing but a cross of two individuals differing in two pairs of contrasting characters. Or we can say, it is a dihybrid inheritance. The dihybrid inheritance is the inheritance of two separate genes, and each gene has two alleles. So I am taking actually as per the dihybrid cross Mendel did. He took two traits, two characters. So what are the two characters? One is called the shape of the seed. Shape of the seed. Another one, color of the characters. Cotyledon. Color of the cotyledons. So these are the two characters they are taken. So I mentioned already it's a cross involving two pairs of contrasting characters. What's the meaning for that one? If we take the shape of the seed, there are two varieties. One is round, another one is wrinkled. So this is one pair of contrasting characters. Now color of the cotyledon concern. Again there are two contrasting characters for the color. One character is normally just yellow. This is the color of the cotyledon. Another one green. That's why I mentioned crosses involving two different pairs of contrasting characters, and they are called dihybrid crosses. Even I mentioned it is the inheritance of two separate genes, and each gene has two alleles. So two separate genes, we have the R gene. One gene is capital R, another one small r. So it is an inheritance of two separate genes, gene 1 R, another one gene Y, gene Y, gene R, gene Y. So these are the two genes, generally orally we can say R gene and Y gene. And these genes have alleles. That means each gene has two alleles. Capital R and small r are alleles, and capital Y and small y are alleles. Now the symbolization is given capital R for round because when a cross is effected between round and green, a monohybrid cross, Mendel obtained only the round shaped, just naturally plants with the round shaped seed. Similarly, when we conducted a cross, a monohybrid cross, by taking the color of the cotyledon, taking yellow cotyledon and green cotyledon, we obtained yellow cotyledon, the F1 cross. That's why the gene capital Y is given for the yellow and small y is given for green after the results obtained. First he did only the monohybrid cross, then only he did what is called dihybrid cross. So taking two characters simultaneous, how far the characters have been inherited. So anyway here we see that one, each gene has two alleles. R gene has capital R and small r. Y gene has capital Y and small y. So anyway, dihybrid inheritance, the inheritance of two separate genes, R and Y genes, and each gene has two alleles. So capital R, small r alleles for R gene, capital Y and small y, the genes for, that is actually Y gene. Took two characters for dihybrid cross. One character is concerned with the shape of the seed, another one is concerned with the color of the cotyledon. And he took parents, one parent with what is called both the dominant characters, that is round and yellow. The genotype for this one, as capital R is dominant over small r, now the genotype for round, that is capital R, capital R. And for the yellow cotyledon, the dominant allele, capital Y, capital Y. While we are taking two genes, normally denotes a diploid condition. It denotes a diploid condition. I will express the same one also with matchstick figures. First I explain with the letters. And another plan he took, that is the shape of the seed, wrinkled, and cotyledon color, green. So it is nothing but actually a cross. It's a combination of two single actually processes. Two monohybrid processes are combined together. Two single actually monohybrid processes, and that results in what we have a dihybrid cross. Now, during gamete formation, there is one allele according to the law of segregation. During gametic formation, there are two alleles of a pair segregate and assort out independently into the gametes. I will explain later what is independent assortment. So, they are being assorted out independently so that each gamete receives one gene for a character. So, for round there is one gene. So, it denotes a haploid condition, hence I mentioned the haploid gametes. So, this is actually haploid gametes having only one gene for a character. So, one gene for round and one gene for yellow. So, all the gametes are similar, having capital R for round and capital Y for yellow. Similarly, the wrinkled green cotyledon pair produces gamete, each with one gene for wrinkled and one gene for yellow, that is small r and small y. So all the gametes are similar. Now, during F1 versus F1 fertilization, this is also the selfing, F1 versus F1 fertilization. So what will happen? Each zygote having equal probability receive one of the four combinations of the gametes from each pair. What I am telling is actually you have to follow the formation of gametes in the next step. So anyway, the F1 generation, because of the union of haploid gametes, one from round yellow cotyledon, another one from wrinkled green cotyledon plants, we are getting what is called a dihybrid. A dihybrid is an individual differing in two pairs of genes. Now this is one pair of genes, this is another pair of genes, hence called dihybrid. In monohybrid, the hybrid is differing only one pair of genes, but here it is differing two pairs of genes. So in F1 generation, we have received a dihybrid. What is the nature of the genotype? Capital R, small r, capital Y, small y. Then the phenotype, round yellow. Now what would happen if this F1, that is dihybrid plants are self? What are the types of gametes produced by each pair? We'll see. Self pair, the F1 generation. See, this is what is called self-pollination or self-fertilization. Two F1 hybrids are taken. One is a female parent, another one is a male parent. And both are having the same characteristics but hybrid or heterozygous. That is round yellow cotyledon. So round shaped seed with yellow cotyledon in both male and female parents. Now this is the genotype. That is the genotype, you see that one. Both the alleles are found in heterozygous condition. So this is one pair of alleles, this is another pair of alleles. Here also this is one pair of alleles, another pair of alleles. Now, during the gamete formation at the time of meiosis, according to Mendel, as the genes are present on different chromosomes, they assort independently of one another. What's the meaning? You see, here the R genes are present, that is what is called the shape of the seed. In 7th chromosome, you know that one. The color of the cotyledon present in chromosome number 1. So they are present in different chromosomes. As they are present in different chromosomes, the one pair of alleles or characters assort out independently of other pair of characters. The meaning for that one, this chromosome with R gene, capital R gene, may combine with what is called capital Y, or it may combine with small y. This is what is called random combination. Or it may combine with, so if you are taking for example, capital R may combine with capital Y or small y. This is what is called independent assortment. There is no connection between the R and then capital Y small y genes, they may combine at any combinations. Similarly, the recessive gene may combine with small y as well as capital Y. So it combines with either capital Y or with small. As a result of this one, because of independent assortment, the meaning for that one, the genes form two pairs of characters are located on two different chromosomes, and during gamete formation they segregate and assort out independently of one another. So the inheritance of these two all genes, that is no way connected to the inheritance of Y genes. Now the OR gene, the dominant gene may combine with dominant Y or recessive one. And similarly, the recessive gene for ringless sheep may combine with capital Y or small y. As a result, we have received four different types of gametes. So this is what we call independent assortment. Capital R may combine with capital Y or small y to form two different types of gametes. Similarly, small R may combine with capital Y or small y, forming two different types of gametes. So the number of gametes produced by a dihybrid is always 4. The number of gametes produced by monohybrid is always 2. If you are taking the trihybrid class, the number of gametes produced by a trihybrid is 8. So the number of gametes produced in each case is increasing geometrically. In monohybrid class, 2 gametes. In dihybrid class, 4 gametes. In trihybrid class, 8 gametes. If you are taking 4 characters, then we are getting 16. So the number of gametes produced by a hybrid, a dihybrid or a trihybrid, they are increasing geometrically. 2, 4, 8, etc. Now what are the gametes produced? So what I mentioned is nothing but the independent assortment. Now let's take the types of gametes produced. I mentioned already, this R may combine with, that is capital Y, or it may combine with small y. These are the 2 gametes. Similarly, the recessive gene R may combine with capital Y to form one gamete, or it may combine with, that is small y, a recessive gene to form another gamete. So here we are getting 4 different types of gametes. So I am writing once again. So capital R may combine with capital Y, or it may combine with small y. Small r may combine with capital Y, or small r may combine with small. This is what is called actually independent assortment of the genes, so that we have different combinations of gametes. But in the case of dihybrid, I mentioned already, 4 gametes will be produced. If it is a trihybrid, we are getting 8 gametes. Now how can you remember? Now these are all F1 haploid gametes. F1 haploid gametes. So during fertilization, what will happen? The zygote, each zygote with equal probability receiver, that is one of the 4 combinations. So during fertilization, that is F1 into F1, each zygote with equal probability receive any one of the 4 combinations from each pair. For example, maybe like this or like this. Okay, any one of the 4 combinations from each pair, so that we are getting the gametes with the different what is called genetic factors. So the gametes are genetically different. This is because of the random union, because of the independent assortment. So now what are the results after? Then how can you remember the gametes form? What is the shortcut method to remember the formation of gametes? Suppose you are taking one, I am taking for example a dihybrid with the letters, any alphabet. So I am talking for example, this is a dihybrid, taking alphabets A and B. This is for one pair of, that is characters, and this is for another pair of characters. One pair of characters and this is another pair of characters. How can you remember the gametes? So the number of gametes produced is actually, we have a formula, 2 power n formula. The n refers to, n is equal to number of heterozygous loci. Here we have So this is one heterozygous loci. This is another heterozygous loci. So here 2 power n is equal to 2 power 2, we are getting 4 gametes. If you have, for example, I'm taking another one, this is a trihybrid. Here just actually 3 different trihybrid, 3 different actually what's called heterozygous loci, 2 power 3. So we are getting 8. So this is one method of just remembering how many gametes are produced. Another way of writing. So how to remember the formation of gametes? You take the first 2 letters. As we know there are 4 gametes are produced. Take the first 2 letters. Write each one twice. I am taking capital B. Write it twice. Small a. Write it twice. Taking the next 2 alphabets, capital B, small b, alternately. So we are getting 4 gametes. This is a method of remembering instead of just memorizing, instead of just a shortcut method of remembering how many gametes will be produced, the formula 2 power n. And also the method of writing in the examination. Take the first 2 letters. Write each one twice, capital A twice, small a twice. Then add the next 2 letters, capital B, small b, alternately. We are getting 4 different types of gametes. Now I will take the same one, apply the same method here. So we are taking round L. This is round L. This is round L. Now I am taking the first 2 letters, twice each, capital R twice, small r twice. Now writing the next 2 letters alternately, capital Y, small y, capital Y, small y. You see the same gametes what we are getting. 1, 2, 3, 4 gametes what we are getting. So this is about the formation of gametes, F1 haploid gametes. If a dihybrid is there, it will produce 4 different types of gametes in equal numbers, not in different numbers, in equal numbers. That is normally found in the case of Mendelian cross. Okay, now, so I mentioned already during F1 fertilization, when F1s are self-fertilized, what would happen? Each zygote normally with equal probability receive one of the 4 gametes from each parent. So we are getting actually 16 individuals. First we are getting actually 4 gametes which are genetically different. By random combination and union of these 4 gametes, 4 into 4, on either side we have 4 different types, 4 into 4, 16 individuals are produced. 16 individuals are produced. And when you are following the ratio, there are 4 individuals produced in the ratio of 9:3:3:1. 9:3:3:1. So this is the ratio what we are obtaining. But in actual practice, we received 556 individuals. 556 individuals, which are being distributed in the ratio of 9:3:3:1. That is what is called actually that is fraction, and this is the total number of individuals produced. So by means of random union, we are getting 16 individuals and we are getting 4 different types of individuals. Now the number of or the types of individuals produced. The types of individuals produced is equal to the number of gametes. This is universal constant. See here there are 4 different types of phenotypes. So we are getting 4 different types of phenotypes. That is equal to the number of gametes produced. If you are taking for example trihybrid cross, there are 8 gametes are produced. In F2 generation we are getting what is called 8 different phenotypes. So anyway, the number of phenotypes produced is equal to the number of gametes produced. See in the case of monohybrid cross, there are 2 gametes. Then the number of phenotypes 2, 3 tom and 1 dwarf, 2 phenotypes. And here we are getting what is called actually 4 phenotypes equal to the number of gametes produced. Now this dihybrid ratio is an ideal ratio. It is based on the probability including the various factors like law of segregation, independent assortment and random fertilization. So it is actually a ratio that is normally including. It's a what is called a proportion. It's a proportion including the law of segregation, law of independent assortment and random fertilization. Okay, now this is one concept. The number of individuals produced or the types of actual phenotypes in F2 is equal to the number of gametes. I mentioned already 16 individuals, that is a fraction, but totally 556 individuals are produced. Now what are the different types of individuals produced? Let's go for the Punnett square. I already mentioned in self-fertilization of F1 plants, each zygote with equal probability receive one of the four, that is combinations from each parent. So this is what we are the gametes of the male parent. These are the gametes of female parent represented actually what we have the graphical method Punnett square. Now it's simple multiplication process. How to remember this one? So this gamete, or we can say this gamete is being fertilized by any one of the four gametes. This gamete, the female gamete, is fertilized by any one of the four gametes. Now I am writing the combination, simple multiplication. This is 1. And this is box number 1, this is box number 2, this is box number 3, this is box number 4 horizontally. I am taking vertically also box number 2, box number 3, box number 4 to identify. Now the second one, equal chance of fertilization, random mating only. Now we are getting, this is another one. Now we are just taking this egg being fertilized by another gamete. This is one. Now in the last column we have capital R small r, capital Y small y. So if you analyze, here you see all the genes are dominant. All the genes are dominant. And also homozygous for each character. So this is homozygous round, this is homozygous here. Now here, 3 genes are dominant. In box number 2 and box number 3, we have 3 genes are dominant. Here, the round is homozygous, the gene for round is homozygous. Here the gene for yellow is homozygous, but the total number of genes, normally dominant genes 3. Now in the last case, 1 dominant gene for round, 1 capital Y for yellow color. So all are round yellow. All are round yellow. 1 dominant R is enough to produce the round. 1 dominant Y is present to produce yellow. Here we have in all cases at least 1 dominant gene is present for the expression of both the characters, both the dominant characters, round and yellow. I am taking just simply, so we are taking this gamete. Similarly, it is being fertilized randomly by all the 4 types randomly. Now for easy observation, now these are the 4 gametes, I put the numbers also in the box. Whatever genotype we are getting in box number 2, here getting in that box vertically also. Capital R, you analyze it later and find out also, work out. Then what you have in the third box, here the same one, the third box. This is simple remembrance, simple method of just keeping in mind. Now whatever you have in the fourth box, here also in the fourth box. Fourth box. Again, if you are taking the diagonal line, now starting from here, here, here and here, whatever you have in the fourth box, here also the fourth box, you have in the diagonal line, the same genotype. So all these genotypes denote at least 1 dominant gene for round and 1 recessive gene for, that is, 1 dominant gene for round and 1 dominant gene for yellow cotton. So if you are counting the total number 1, 2, 3, 4, 5, 6, 7, 8, 9. So we have 9 individuals round yellow. This is 1. Now the next one, new combination. Combinations we have 3. And all the 3 are round green. Round green. So I am getting 3 individuals round green. So now I am writing here, this is round green. Both the genes are homozygous. And here I am writing, just you see that one gene is heterozygous and one gene is homozygous. The same one what we are getting here also. So just one with both homozygous, and here one is homozygous, another one is heterozygous. Here one is heterozygous, another one is homozygous. These 3 are what is called round green. Okay, this is another one. Let's go for another one. Another combination we have 3 wrinkle yellow. 3 wrinkle yellow. So we have wrinkle character. Normally you see that one. This is wrinkle yellow. Both the genes are homozygous. Now here only one gene small r is homozygous. Another gene is heterozygous. Another gene is heterozygous. So these 3 represents what we have the wrinkled yellow. See, wrinkled necessity yellow, at least one dominant gene. Here 2 dominant genes, here at least one dominant gene is present. The last one, what we have, one wrinkled green. One wrinkled green. So we have both the genes are recessive. Both the genes are recessive. So that one indicated, as we have 4 different types of gametes, also we are getting 4 different types of phenotypes in the ratio of 9:3:3:1. Now, normally he got actually 4 types of individuals. This is the fraction, the ratio, you see that one, fraction. But totally he received 556 individuals. 556. And one we have that is round yellow. 350. Round green. 108. That is wrinkle yellow. 101. Then wrinkled green. So we have 32. Total of 56. So this is the total number of individuals he received. He made it into fraction of 9:3:3:1. That ratio what is obtained in F2 generation is called the phenotypic ratio. Phenotypic ratio of 9 round yellow, 3 round green, 3 what is called actually wrinkled yellow and 1 wrinkled green. This is called the phenotypic ratio. How can it? So in the book given what is the stick figure, I will follow that one later. Just how to find out the answer for this one. Now, if you are taking these, the black, and drawing a line, that is a triangle, a big triangle. These individuals in the big triangle represent round yellow. Then we are taking another individual that is round green. These 3, you see that one, present apparently or present at the 3 corners of the apparent what we call triangle. Then these 3 individuals are present in a small triangle. That is what is called wrinkled yellow variety. Now the last one, see, this is the last one. Only one at the corner. Now if you are analyzing what we have the genotype, now taking, see, just this one. Now I am taking this diagonal line, and in each case, only one individual out of 9 round yellow, only one is homozygous for both the genes. And here you see that one for round green, only one out of 3, only one is homozygous for both the genes. Here in the case of wrinkled yellow, only one is homozygous for both the genes, others are normally not. Just completely homozygous. Here all the genes, both small r and small y, are just in homozygous condition. This is a simple method of just remembering that one. Okay, now, so this is the phenotypic ratio what we obtain. Now, out of what is called 16 individuals produced, how many of them are parental combinations? We have received parental as well as non-parental combinations. What are the parental combinations and non-parental combinations? The parental combinations are nothing but the parental varieties what we start for the experiment. We started the experiment with the round yellow and wrinkled green. These are the parental combinations. So, out of the 16, now this round yellow, this is one of the parents. Another one just actually wrinkled green. This is another parent we started. So, parental combinations are totally parental combinations. So, we have 9 plus 1, totally 10. So, 9 round yellow, 1 wrinkled. They are the parental combinations. Now, the new combinations. The new combinations is that one round, instead of yellow we have green is present. Now wrinkled, instead of green, yellow is present. These two are called actually new combinations. 3 plus 3, that is equal to 6. So, the ratio between the parental combinations and new combinations is equal to 10:6. This is the ratio between the parental and then what we have the new combinations. And we can reduce further also 5:3. 5:3. Now this is about the parental combinations and new combinations ratio, that is referring to the parents as well as what we have, that is the new combinations. Now we have made what is called the phenotypic ratio. Is there any genotypic ratio? Is it possible to find out the genotypic ratio in the case of such organisms? Students, now let us analyze the genotype of each phenotype. I am taking the first phenotype round yellow, totally 9. Though they are externally similar, their genetic constitution is different. There are 4 different types. Number 1, so this is one genotype, this is another genotype, this is another genotype, this is another genotype. So I am writing here capital R, capital R, capital Y, capital Y. The next one, capital R, that is capital Y, small y. The next one, this is what we have. Capital R, small r, capital Y, capital Y. The last one we have. So in the first case, 2, you see that one, 4 dominant genes. The second case, we have 3 dominant genes. The third case, we have again 3 dominant genes. The fourth case, only 2 dominant genes. Now what is a number? So the first one is present 1 genotype. The next one 2. You see that one here and here 2 genotypes. The next one is also 2 in number. This one and this one. And the next one is 4. Totally 9. So the genotypic ratio 1:2, 2:2, 2:4 for round yellow. Now I am taking round green. Round green, there are 2 genotypes. This is one genotype, this is another genotype. So capital R, small y, and then capital R, small y. So 2, 1:2. If I am taking the next one, wrinkled yellow, I am writing here wrinkled yellow. Again here there are 2 genotypes. One with what is called actually small r, capital Y. So this is 1. Small r, capital Y, small y. This is 2. See that one? This is one type. These 2 are another types. So the ratio 1:2. And the last one, wrinkled green, only 1. So if you are taking the genotypic ratio 1:2. 2 is to 2 is to 4, then 1 is to 2, 1 is 2, and then what we have 1. So this is what we have 9, this is what we have another type 3, and this is we have another type 3, and this is 1. So we are getting, you see that 1, the phenotypic ratio 9 is to 3 is to 3 is to 1. For dominant 9, there are 4 genotypes. For the next type, round green, 2 genotypes. The regular yellow, 2 genotypes. For the recessive one, only 1. So totally you can get, you see that 9 genotypes. Number of phenotypes is 4, but the total number of genotypes is equal to 9. How can you remember? What is the maximum number in the phenotypic ratio? That is 9. So we have 9 genotypes. If you are taking the monohybrid cross, 3 is to 1. That's the phenotypic ratio. But the number of genotypes 1, 2 and 3. Now the maximum number in the phenotypic ratio is 3. And now the number of genotypes 1, 2, 3. This is the easy method of remembering, that is a genotypic ratio. For example, if we are taking trihybrid cross, the ratio is 27, 999, 333 and then 1. Now there are 8 phenotypes. The number of phenotypes 8. Now how many genotypes? If you know the maximum number for monohybrid cross, the genotypes 3, the first number. If you are taking, for example, dihybrid cross, the maximum number in the ratio 9. So the number of genotypes 9. So without calculating, one can say the number of genotypes in trihybrid cross is 27. That is the first number, which is maximum, is corresponding to the number of genotypes. I want to represent the dihybrid cross, what I discussed earlier, in the form of stick figures as given in the book. So we are taking the parent generation, parent 1 the female, parent 2 the male. The parent 1 is round with yellow cotyledon, and the parent 2, that is the male, we are talking about wrinkled green cotyledon. Now this is what we have the genotype, represented in the form of chromosomes. You know that one, the gene for the shape of the seed located in chromosome number 7, and the gene for color of the cotyledon located in chromosome number 1. So that is about the location. So this is round yellow, that is about the genotype represented chromosomes. Now this is wrinkled green, and here too you see that one, the gene for R is located, the shape of the seed is located, as I mentioned there, you have 7, this is 1 color. Now, they produce the gametes, haploid gametes. So, independent assortment, segregation. So that this is a gamete with capital R, capital Y, and this is a gamete produced by what? The parent wrinkled green cotyledon, smaller Y. Now, gametic union by means of fertilization, we are getting F1 generation. That is, it's heterozygous. That is round yellow. So, one capital R, one small r, one capital Y, one small y located in the chromosomes. The red line indicates the location of the genes. Now, F1 generation, this is heterozygous dihybrid. Now, when F1 generation itself, I represent only this way, F1 into F1, that is male and female. Now, 4 haploid gametes are produced by each parent. Now, I represent the 4 haploid gametes, one with capital RY, one with capital R small y, one with small r capital Y, another one with a small r y. Okay, this is the arrangement in both male and female gametes. So during fertilization, each zygote with equal probability receiving one of the 4 combinations from each parent. So we are getting just as in the case of previous illustration, what I represented with the letters, here represented just in the form of stick figures. In all cases, we have to represent actually the chromosome containing the gene for the shape of the seed. Here it is represented by blue color, and the another one where you have represent the gene that is red color one. In the case of YG, that is in the chromosome, just normally representing the color of the coordinate, that is 1. So as a rule, we are getting actually see that 4 types of individuals. Now this is one case, what I mentioned already, 9 round yellow, this one, 9 round yellow. Then we are getting just we have 3 round green. So this is round green individuals, 3 round green. Then we have 3 wrinkled yellow. Now this is wrinkled yellow. This is what is how wrinkled yellow. And finally only one, just what we have with reference to the wrinkled green, both characters being recessive, represented by the gene small r, small y. So the phenotypes we are getting as mentioned earlier, 9, 3, 3, 1. The phenotypic ratio 9:3:3:1, what I described already. So you have to represent this way as it is represented in the book in the examination, writing the letters along with the chromosomal structure. Okay, so this is the one what I talked about, the dihedral cross. I represent both the way, either in the form of letters or in the form of, that is, majestic figures showing the chromosomes. Thank you.